Step up. Great. Welcome to Colloquium. I'm Jake Brunkard today for the purposes of this colloquium. So I'll be introducing Erin Alberts here. Erin trained as a microbiologist at Oregon State, investigating microbial effectors in Pseudomonas strains. And when Erin arrived in Madison, she started a new direction in the Brunkard Lab, investigating how target of rapamycin, or TOR, and its interacting partner Raptor coordinate growth defense trade-offs. For this project, Erin received an NSF Graduate Research Fellowship, and today, Erin is going to tell you about a necessary detour she's taken to address an outstanding mystery in the plant TOR literature. Are Raptor mutants lethal or not?
Thank you. Hello, my name is Erin. If anyone saw the posters that were printed, this is not what is on the posters. I was originally going to present that, but then I thought that this would be a more interesting story to go over. So we're going in a different direction, still focusing on TOR and plants. I just have to click on it. Yes. Thank you. All right, some general background on TOR, or target of rapamycin. TOR is an evolutionarily conserved serine threonine kinase that's present in all eukaryotes, from protists to humans to mice. TOR acts as a nutrient sensor that integrates upstream signals like amino acids, nitrogen, phosphorus, things that affect circadian rhythm as well, like sunlight, nighttime, temperature, and regulates growth and development. Some of those major outputs are regulation of translation, ribosome biogenesis, primary metabolism, and autophagy. TOR itself functions as a part of the TORC1 complex, which looks like this. It's composed of 3 parts, which we're going to go through in the next slide. Oh no. Okay. The slide is wonky, but what it says is TORC1 is a dimer comprised of TOR, LST8, and Raptor protoheterotrimers. And each of these 3 protoheterotrimers has its own role in the TORC1 complex. LST8, or lethal with Sec13-8, functions as a scaffold for substrates. Left is the cartoon, right is the protein structure. Yes, probably guessed. But I've circled the same component in both the cartoon and the protein structure. TOR is the catalytic subunit. That's where the kinase activity actually takes place. And Raptor recruits substrates to the overall complex. The TORC1 complex must function as this dimer. The separate components don't really have much activity on their own. One thing that's particularly interesting about TOR and the TORC1 complex is that it's highly, highly evolutionarily conserved and was found in the last eukaryotic common ancestor, which was about 2 billion years ago. As you can see on the top there, I'm going to use the laser pointer, TORC1. And then later, TORC2, which is only found in mammalian systems, evolved as well as many other things. But TORC1 includes Raptor, LST8. All of those things are very highly evolutionarily conserved. Therefore, it rather goes without saying that Raptor is highly evolutionarily conserved. This is a comparison of the different Raptor proteins between humans and flies and yeast and C. elegans, and then Arabidopsis down here at the bottom. You can see very similar. We have the RNC domain, which is where we think most of the activity happens. This over here is the WD40 domain, which no one really knows what it does, but it appears to be essential for the function of Raptor. And one of the reasons that I particularly like this diagram is that it shows how closely related the Raptor proteins are between humans and Arabidopsis. So one of the things that our lab does is sort of uses plants as a model system and relates that to human health. Funded by the NIH. And you can see they're very close together. This is another just little diagram that shows the different Raptor orthologs in yeast and rice and mammals. again, very closely related to each other. And some people in this room will know one or other of these different genes. All right, so now we're going to switch into thinking about Arabidopsis solely. There are two raptor homologs in Arabidopsis thaliana. This is very confusing for everyone. Raptor 1b, or raptor 1, is highly expressed throughout development. It's expressed in the plant everywhere all the time, in the leaves, the roots, the shoots, basically every plant tissue, and throughout development. So from the embryonic stage to adult plants. Raptor 1a or raptor 2 has lower levels of expression throughout development, and it is mostly found in leaves and flowers and not so much in roots or seeds. So that would kind of lead one to believe that it has roles in later development, not so much in early development. If you're wondering, raptor 1a and raptor 1b have an 83% sequence similarity. Right. So now we're going to sort of move on to the question that I'm trying to answer, which is about the function of raptor in the TORC1 complex and how essential it is for early growth in Arabidopsis. One of the reasons that we even ask this question is that we know that TOR mutation is embryo lethal in Arabidopsis. On the top, you can see, well, on the very top, you can see a salique from Arabidopsis. These white seeds are the raptor or the TOR mutant embryos. They're white. They are very sad. Do not look right. These are normal embryos going through the stages of development. And then on the bottom, we have TOR mutant embryos. So these are complete TOR knockouts. And as you can see, they don't get past what is called the preglobular stage of embryonic development. They are not going to grow past this point. Now the question comes in whether or not raptor 1b mutation or complete knockout may or may not be embryo lethal. I'm focusing on raptor 1b right now because it's the higher expressed of the two genes. And again, it's expressed in all tissues all the time, rather than just sort of in the later stages of plant development. There are a bunch of papers, and I'm going to show a few of them, that sort of give us conflicting results, conflicting opinions about whether or not raptor 1b knockout is or is not embryo lethal. So in 2005, these two papers came out around the same time. This Anderson paper showed that a raptor 1b mutant was viable and could produce a viable plant. There were definitely growth defects. As you can see, it's a lot smaller. It's a lot wimpier. And then this other paper came out the same year that showed that, I don't know if this is a super clear figure, but you can see this little tiny raptor 1 seed, which is raptor 1b. Not good, not gonna grow, very much embryo lethal, like will not produce a plant. They found that disrupting raptor 1a doesn't lead to any phenotype. They used two different lines here, which I'm going to get into later and as part of hypotheses, but essentially they both think that these are raptor 1b mutants. The people that wrote this paper think that this difference is due to a difference in growth conditions, basically saying that the DePros lab like stressed out their plants, and that's why these seeds didn't grow. I don't know about that, but that's what this is about. This is the mystery we're trying to answer. In subsequent papers, they also were able to produce raptor plants that were viable. This is a study about abscisic acid, but as you can see, they also used two different lines. One of the things that was strange, these two, one of the things that was strange to me about this is that they had a higher germination rate for one than the other, which sort of leads me to believe that if you think that they're both knockouts of the same gene, why is one germinating more than the other? But we'll let that go, I guess. They also found, so it's sort of generally known as an aside that LSTA is not lethal when you knock it out. And then, even later days, even bigger plants from the raptor knockouts, they're using the same lines as before, and somehow the plants again look slightly different and even more hardy. As the same with this paper, I think these two use the same line These people suggested that the knockout of Raptor 1b was partially compensated for by the increased expression of Raptor 1a, which again is something that is very possible, and we'd like to figure that out if that actually is occurring, because that would again be something that we don't know about yet. So all these mutants have different phenotypes, and they're all purported to be Raptor 1b knockouts. So first thing to do is take a look at how they were created, to see if we can figure out maybe some differences between the different lines and how they were actually generated and obtained by the labs. So the way that these lines were generated was via tDNA insertion mutagenesis. tDNA stands for transfer DNA, and it is located on the Agrobacterium Ti plasmid, which looks like this. It's this region right here. And the way this works is that Agrobacterium injects its genomic material into a plant cell. It basically gets integrated into the plant genome. And so you have these big chunks of tDNA that are inserted into your plant genome, and this supposedly disrupts the transcription and translation of the gene, leading to a knockout. The lines that are used in all these papers were generated in 2003 by a random tDNA mutagenesis, in which all of the lines were then sequenced and the insertions were mapped. And these are the lines that like almost everyone uses today. But that's a little background on that. Cool. So one of the hypotheses we have about these lines is that the Raptor 1b tDNA insertion lines may not actually be effectively abolishing Raptor 1b function. This was my theory, that because these insertions are all sort of in the upstream like section of the gene, that potentially, even though there is a big chunk of tDNA in there, that potentially this part is being transcribed, or like this one, maybe the rest of it's being transcribed, or they're just not actually effectively abolishing the function of the gene. We really don't know that much about how tDNA insertion affects the functioning of genes. There are many weird things that happen with these sorts of lines, like inversions and relocations and weird things. So I don't think it's enough to say that because we have a tDNA insertion in this gene that it's knocked out for sure. I can tell you what all the different papers are that use these different lines, but basically they all used different Salk lines, which is the institute that created the lines in the first place. Another reason that we think that this is the case, besides my own thought process, is that our lab did a multi-omics analysis on Salk 078159, which is this one right here, that a lot of these papers used in their studies, which is Raptor 1.1, generally referred to. And it found that in the transcriptome, there was expression of Raptor 1b, in the proteome, that there were Raptor 1b peptides present, and then in the phosphoproteome, which is the way that we tell whether TOR is active or not, because it phosphorylates things, there was phosphorylation of ES6, which is one of TOR's downstream substrates. So this would suggest that potentially this line is not actually abolishing Raptor function, even if it's maybe messing with the gene. Raptor can still function, and therefore these lines would actually not be Raptor 1b knockouts, but maybe just like slightly less effective mutants. I thought I would show this picture of the Raptor protein with this other little picture that I made. So you can see sort of where these insert in here. Again, don't know what the WD-40 domain does. The heat repeats are where it binds to the TOR protein, and then the RNC domain is, I believe, where the function is. So this is just kind of a theory, but it's worth testing, I think. So one of the ways that we can test this is to actually create a real, full, complete knockout of Raptor 1b, potentially Raptor 1a, homozygous mutants. And the way that I'm going to be doing this, the way that I have been working on doing it, is by using a Cas9 Act 3.0 CRISPR combo system that was recently developed by, what's his name? Yiping QI, his lab, and it enables some very interesting gene editing approaches. It enables transcriptional activation as well as editing. So on the top here, we have well, if you use this particular 20 nucleotide protospacer, it leads to double-stranded breaks, just like sort of a normal CRISPR system. But if you use a 15 nucleotide protospacer with these MS2 RNA aptamers that recruit these sun tags, you can generate transcriptional activation, which helps with the next piece of this, which is that if you add flowering locus T into this gene activation section of the plasmid, you can more easily screen for mutants, which reduces the time to finding mutant raptor or mutant whatever plants you want to get. So you put in flowering locus, which leads to early flowering. Then you put in your raptor gene in the other part of the plasmid. You can multiplex these together. Synthesize oligos. This is just sort of methodology stuff, but you basically multiplex all these together, and then you add them into your Cas9 Act 3.0, and then put this all into your destination vector, and then you construct your CRISPR vector that you eventually put into your plants through agrobacteria-mediated floral dipping, which is the same process as I was showing you before with the TI plasmid. So you do this dip, and then where that interesting gene activation part comes in is that in your plants that have this CRISPR plasmid in them, they have very, very early flowering, and that's how you can identify which plants have been successfully edited versus ones that haven't. And so these plants essentially will have your edit in them as well as that early flowering gene. So then you extract your DNA, you do your genotyping, and then you raise the progeny of these to screen for transgene-free plants. So you don't want the CRISPR transgene still in there. And then you get your mutant plants, hopefully. So if my hypothesis is right, which is that the Raptor1b mutant, double mutant, would be embryo lethal, essentially I would be getting Raptor1b heterozygotes, which I would self. I would then harvest the seeds and look for these, like we saw before, these sort of like white, transparent, like very sickly looking seeds, separate them from these, and from the seeds, extract RNA and protein, and then do some other assays down the road. But really, I'm interested in this part, which is figuring out whether or not these seeds are actually viable. I've been working on optimizing all of these steps and also making this procedure a lot easier for undergraduates because of this sort of like modality where you could essentially synthesize any oligo that you want, put it into your CRISPR plasmid. Any plant gene that you want to knock out should essentially work with the same system. So I've been trying to optimize that potentially as things for maybe newcomers into the lab or undergrads to work on as like a good project. And CRISPR is so hot these days. Everyone wants to do it. All right. I think that's the end. Cool. And then this is the Brunkard Lab. I also don't have a picture of KD2, but this is most of the Brunkard Lab. Thanks to the NIH and the NSF and the USDA. And questions? If anyone has questions, feel free to ask them.
Great talk. I had a question about, like, you showed like the map with the 4 different raptor DNA insertions. I was curious, like, for like the people that published their papers in 2005, like which one, like, do you know which one was like the one that was the lethal one?
Yeah, I do know. And I have it written down. Oh, it was, it's the ones that aren't on here. Because when I made this figure, I didn't put them on here. But one of those insertions is like right here. And one of them is like right here.
So like, do you know which one the lethal one is? Cause that's really interesting.
I think it was either, I don't actually know. Because I think both of them were when they used them. But it was like these areas right here.
When you guys did the multi-omics,
Mhm.
was there any indication that there was reduction of that? Like, and I'm sure you guys did it, but you also just did a regular genomic PCR to make sure the insert was really there, right?
Yeah. Mhm.
But was there any indication that was any loss of?
Now that you say that, I think there was. I think there was like a small like reduction in the expression, but it wasn't enough for me to say that was actually a knockout. Because I remember looking at it and being like, it's a little less than wild type, but not so much that you would think that that was like a knockout of the gene.
It might be a big.
Yeah.
And you guys don't have deletion with the other dogs like that.
They're harder to make. Yeah, I wish. Yeah. Oh.
There's two problems. Do you think that they wanted to spread? I mean, I guess you think it is a compensation. So they're only expressing six cell type, but it could be reacting to different cell types or different processes.
We actually sort of thought about this, and we kind of think that because all of this stuff is so old, like I was saying, there's like, it's very evolutionarily, like a long time ago, that is actually sort of going through this process of being lost. That potentially in the past it had higher expression, but now it's like getting lower and lower and lower and lower, so that it's actually like, I don't know what that's called in evolution when something is sort of like going away, but that's what it looks like based on what we looked at, the expression and all of that. So, but it's still able to compensate a little bit.
Cell level that you can visualize whether different cells are expressing.
I do not.